So greetings, everyone, and welcome to the Science of Childhood Cancer lecture series that's hosted by the St. Jude Children's Research Hospital Comprehensive Cancer Center. I'm Charlie Roberts, Cancer Center Director and Executive Vice President here at St. Jude, and I have to say it is my true pleasure today to welcome our speaker, Dr. Alejandro Gutierrez. I've known Dr. Gutierrez for a number of years from my time in Boston, and it's a real pleasure to host him. And so I'm really pleased now to introduce Dr. Gutierrez. So he did his undergraduate work at the University of Arizona in chemistry. From there, he went to the University of Pennsylvania School of Medicine for his MD degree. Following that, he did a residency in pediatrics, staying on in Philadelphia at the Children's Hospital of Philadelphia. And from there, moved to Boston to do his fellowship in pediatric hematology oncology at Dana-Farber Boston Children's. He chose to do his postdoctoral research work in the laboratory of Tom Look at Dana-Farber, and then launched his own laboratory, becoming an assistant professor at Boston Children's Hospital. And last year, he was promoted to associate professor. And I think a nice reflection of the impact of his work. He was also given an endowed chair, the Louis K. Diamond Chair in Hematology Oncology at Boston Children's. Dr. Gutierrez has some notable honors, including being elected to the Society of Pediatric Research, elected recently to the American Society for Clinical Investigation. He's also had a Clinical Investigator Award from Damon Runyon. And I think really telling of the Alejandro I know is that he has numerous awards for excellence in teaching, excellence in mentoring. I think it really reflects the passion that he has in developing the careers of others. And he's also been the recipient of the Stephen Sallin Leadership Award at Dana-Farber. He's well-funded, 2 R01s, a $250,000 grant from Alex's Lemonade Stand Innovation Award, and a $500,000 award from Astellas Pharmaceuticals. He's had numerous high-impact publications and talks, and I think you're going to get to hear about that great work now. And so with that, I'll throw it to you, Alejandro.
Thanks very much, Charlie, for the very kind introduction. This has been a really terrific seminar series, and it's a real honor to be a part of it. And please jump in if you can't see my screen. So here are my disclosures. So I thought I'd begin today with an overview of the published work in this area, and this topic of asparaginase resistance. This is a work that really was initiated by Laura Hinz, a terrific medical student at the time in my lab. And I'll spend most of the talk on this this next story here, which is fairly mature and I think has uncovered what was a previously unrecognized cellular response to amino acid starvation that explains the unique role of GSK3 alpha in asparaginase resistance. I then hope to get through 2 stories that are still works in progress, and certainly they're not complete, but I'd be very eager to get feedback on both of these. And these are work of both Laura and Yunchul here at the end. So as most of you know, the anti-leukemic enzyme asparaginase has a major role in frontline therapy for acute lymphoblastic leukemias. This enzyme degrades the amino acid asparagine, which is considered a non-essential amino acid. This is a dietary definition. In other words, it's not required in the diet. It is, of course, however, required for protein synthesis. And so I think of this as a bit of a misnomer nomenclature. We've known for a long time that many leukemias require access to extracellular asparagine to survive in order to repress cell death. And we, of course, exploit this vulnerability clinically with asparaginase, which depletes circulating asparagine and thus triggers cell death in drug-sensitive leukemias. Using an unbiased genetic screen, Laura found that resistance to asparaginase can arise by a reliance on protein degradation through the ubiquitin proteasome pathway, which provides a catabolic source of amino acids, including asparagine, of course, that allows cells to survive during starvation of asparagine from the environment. This adaptive response is strictly dependent on the catalytic activity of GSK3, and it's really GSK3 alpha and not beta that'll be important for most of the talk. GSK3, it turns out, can phosphorylate a large number of proteins in a cell and typically results in the formation of a phosphodegron that marks these proteins for protein ubiquitination and then degradation through the ubiquitin proteasome pathway, or UPS. And we discovered this pathway because it's blocked by activation of Wnt ligand signaling, or this can also be achieved with a pharmacologic small molecule GSK3 alpha inhibitor. And combining a GSK3 alpha inhibitor with asparaginase has potent activity against patient-derived leukemias that were refractory to monotherapy with either agent, as you see here. And one of the reasons we're so excited about this pathway, and we spent a lot of time on it, is because the effect size here. I'll just draw your attention. This is 12 days of treatment, resulting in really pretty striking, at least in my biased opinion, a pretty striking effect size that I think has real potential to turn into a clinically meaningful patient benefit. So after doing this work in leukemia, Laura then turned her attention to colon cancer. You might ask, why would we think of asparaginase in colon cancer? It turns out that colon cancers, as most of you know, are characterized by activating mutations of the Wnt pathway. But these mutations are not all the same, and we thought that the occurrence of mutations that activate Wnt signaling at different levels of the pathway would provide us with a unique opportunity to test predictions from our model. So most patients with colon cancers have mutations of either APC or beta-catenin, and we predicted that these would selectively activate beta-catenin without blocking GSK3 or disrupting the ability of these tumors to tolerate asparaginase. By contrast, about 15% of colon cancers are driven by mutations of Wnt ligands or R-spondin ligands, or of the R-spondin receptor RNF43. These mutations also activate Wnt beta-catenin signaling, and I would bet my mortgage that they're selected for during colorectal transformation because they activate beta-catenin. But they do so by inhibiting GSK3. And so the idea is actually GSK3 alpha may be inhibited in the tumor cells as a consequence of the oncogenic mutation, and this may prevent these tumors from tolerating asparaginase through this pathway we've uncovered. So we engrafted mice with mouse genetically engineered colon cancer models that were designed to be isogenic except for the type of Wnt pathway mutation. And as you see here, APC mutant tumors, which I'll remind you, these are the cases that we predicted would only activate beta-catenin without impairing asparaginase response, in fact, are completely refractory to asparaginase monotherapy in vivo. But the R-spondin fusion tumors driven by these mutations that inhibit GSK3 are highly sensitive to a single dose of asparaginase. So I also, for the clinicians in the audience, I thought I'd spend a minute thinking or discussing how we're thinking of taking these findings to the clinic. And so there are really two different strategies needed depending on the tumor genotype. So for the rare subset of human cancers driven by mutations in which the Wnt pathway is activated by inhibition of GSK3, in these cases, GSK3 is already blocked in the tumor cell, and so we just need asparaginase therapy. So we've shown this for R-spondin fusion colon cancer. It's about 2% of colon cancer, which is still a lot of patients. And we predict activity in cases that have mutations of the R-spondin receptor RNF43, although don't have direct data on this yet. But this is a higher fraction of adults with cancer, mostly adults. And a clinical trial is under development with collaborators to test this question, hopefully in the relatively near future. Now, for most patients who lack the upstream Wnt activating mutation, we actually need a combination of asparaginase with a pharmacologic GSK3 inhibitor. And we have shown that this works in acute leukemias, in colon cancer, and I predict this should work in other tumor types. You know, there's not a lot of similarities between leukemia and colon cancer that would distinguish them, at least in my mind, from other tumor types. So this may be more broadly relevant. But to exploit this, we really need a GSK3 alpha inhibitor that is clinically usable. So we're making a couple of different pushes on this front, and I hope to have a drug, although that's not imminent. All right, so then I'll move to the new part of the talk. And so this first story is, again, work of Laura. And she was interested in the question of, you know, why is it that asparaginase resistance is so uniquely dependent on GSK3 alpha and not beta? This is surprising because, as you probably know, these enzymes are closely related paralogs, and they're thought to be largely redundant for their biological functions. In fact, loss of a mouse knockout of GSK3 alpha is viable and fertile because GSK3 beta can compensate. So to understand the paralog selectivity here, we began by comparing the protein sequences of these enzymes. As you can see, the kinase domain is almost identical. This domain is also responsible for recruitment to axin and to the beta-catenin destruction complex. And so homology here probably explains why these are redundant for beta-catenin regulation. By contrast, the C-terminus is similarly sized, but not well conserved at the amino acid level. And the N-terminus is most different, with GSK3 alpha having a much longer N-terminal tail that is largely absent from beta. So to understand which features of GSK3 alpha are required for asparaginase resistance, Laura undertook a domain swap approach. So the model is in a T cell ALL cell line that is resistant to asparaginase at baseline. We knock down GSK3 alpha here with an shRNA and we induce sensitivity, so the cells die. And the toxicity of the knockdown is completely rescued by restoring expression of a cDNA encoding full-length GSK3 alpha. And importantly, not beta. Beta is completely dead in this assay. And so now Laura came in with some domain swap experiments to define which of these domains are important. She found that if you take the N-terminus of GSK3 alpha and fuse to the kinase and C-terminal tail of beta, you end up with a GSK3 allele that is completely competent to mediate asparaginase resistance. By contrast, if you take GSK3 alpha, take off its N-terminus and replace it with that of beta, now you've killed its ability to induce asparaginase resistance, and the other domains were interchangeable. So from this, we conclude that the N-terminus of GSK3 alpha is uniquely required for asparaginase resistance. So, of course, what is this N-terminus? It had no described function. If you look at evolutionary conservation, this is highly conserved throughout mammalian evolution, as you see here. Strikingly, though, this is not conserved in other vertebrates. And this maybe is a topic we get into in the Q&A section. You know, why would mammals care so much about asparagine in a way that other vertebrates, other eukaryotes really don't? So, you know, when you try to find a function of a protein domain, there are several traditional approaches. You can apply domain prediction algorithms. You can apply blast approaches because, as you know, during evolution, evolution typically reuses functional protein domains. But we found actually no detectable homology to any other proteins or protein domains, which was surprising for a functionally important protein domain. However, we found an algorithm that predicted actually this domain was actually a low complexity domain. This is called a SEG. SEG is the name of the algorithm that pulled this out. And if you look at the sequence just visually, you see that actually this is highly enriched in glycines and also to a lesser degree prolines and serines. And this results in an amino acid composition that is much less complex than you would predict by chance. And you may know that these low complexity protein domains have been implicated as mediators of biomolecular condensation or liquid-liquid phase separation. This is an increasingly recognized feature through which cells can evolve mechanisms to promote the catalytic efficiency of multi-step enzymatic reactions by concentrating their enzymatic components in physical concentration of these enzymes. So we asked whether GSK3 undergoes physical relocalization in response to asparaginase therapy. And as you can see here, this is GSK3 alpha in red and DAPI's labeling the nucleus in blue. And upon asparaginase therapy, we see a relocalization of GSK3 alpha from a relatively diffuse location into these distinct cytoplasmic puncta. GSK3 beta really does not do this at all unless you swap its N-terminus with that of alpha. And the data are quantified here. So I'll draw your attention to the scale here. So we're counting number of GSK3 alpha bodies in 50 cells. So there's approximately one body per cell, but actually there's some heterogeneity you may be able to appreciate here with some cells forming multiple and other cells forming none. And I'll get back to that heterogeneity later in the talk. So it turns out that formation of these bodies is induced by asparagine depletion. And the first evidence we had for that is that this is a selective response to therapy with asparaginase and this is not just a global response to cytotoxic therapeutics. Moreover, if we take leukemia cells and culture them in the absence of non-essential amino acids, this is in standard media depleted of all, without any non-essential amino acids. Importantly, essential amino acids are present here. this is actually sufficient to trigger formation of these bodies. And if we add back only asparagine, we block their formation. And by contrast, glutamine, which you may know is a physiochemically similar amino acid, actually is unable to block formation of these bodies, and quantified here. So this suggests that at least among non-essential amino acids, depletion of asparagine is unique in its ability to trigger formation of GSK3 alpha bodies. Again, back to this question, why is asparagine so special? So what are these GSK3 alpha bodies? So we first wondered whether this might reflect, well, maybe GSK3 alpha is moving into autophagosomes or some other organelle that you would predict to be formed or to be upregulated in response to starvation. And we found no evidence for this. So for example, we do see some formation of autophagosomes in response to asparaginase therapy, but there's no colocalization at all with GSK3 alpha, or no consistent colocalization. And likewise, we saw no evidence that these were lysosomes or endosomes. You may know that there are also several membraneless cytoplasmic bodies that have been described that are thought to form through liquid-liquid phase separation. And we found that these were not any of these known bodies, at least within the limits of the markers that we assessed. But then we asked, well, we got at this because we're interested in understanding this pathway of protein degradation through the ubiquitin proteasome system. So could these bodies actually function in the UPS? And so this is the pathway I showed you before. And so we looked at different markers that we predict would assemble in these bodies if bodies were assembling the whole machinery for protein degradation through this pathway. In fact, we saw that GSK3 alpha co-localizes with ubiquitin, as you can see here. And this is really strong and consistent co-staining of these proteins. And this is again in response to asparaginase. If we treat cells with a small molecule enzymatic inhibitor of GSK3 alpha, we don't block its ability to undergo spatial sequestration in these bodies, but we block the appearance of ubiquitin in these bodies. And so this suggests that these bodies may be functioning in protein ubiquitination. The data are quantified here. It's easier to see from the pictures than to walk you through the quantification, in my opinion. So what else would you predict might be in these bodies? Well, the proteasome itself. And in fact, we see evidence of co-localization with the proteasome as assessed by immunofluorescence for this proteasomal PSMA4. And you can see that again, we see very nice co-staining of the proteasome with GSK3 alpha, quantified here. So what else might be involved in these bodies? So it turns out there's a large literature, mostly in yeast, about a whole machinery that has evolved to handle misfolded or damaged cytoplasmic proteins. And heat shock proteins are actually important for this. They're involved in unfolding these and targeting them for degradation through the ubiquitin proteasome system. So we asked whether heat shock proteins are involved. And in fact, we found that HSP70 in particular is important here. So we see that in response to asparaginase, we see co-staining of HSP70 with GSK3 alpha. So HSP70 is moving into these bodies as quantified here. Moreover, if we inhibit HSP70 with either two structurally distinct small molecule inhibitors or with an shRNA, we both block formation of bodies entirely, and we also block asparaginase resistance, or we induce sensitivity to asparaginase, indicating that that HSP70 is both required for formation of these bodies and required for asparaginase resistance. So we then asked whether asparaginase therapy induces proteasomal protein degradation in response to formation of these bodies. And so to do this, we relied on a pulse chase experiment with radio-labeled phenylalanine, so it's sort of a prototypical amino acid that is classically used in these assays. So the experiment here is we're pulsing with phenylalanine, and then we're measuring the rate of release of radioactivity during the chase into the media, which indicates protein degradation. And the way you assign this protein degradation to the proteasome is it's defined by being suppressible by bortezomib. And you can see here that at 12 hours of treatment, we see an increase in total protein degradation in asparaginase-treated cells. Now, you might argue, and I this struck us also as a bit of a modest effect, given what we're proposing is a global change in protein degradation. But I'll draw your attention to the fact that at 12 hours, we're only barely starting to see formation of GSK3 alpha bodies. And we speculate that if we were able to do this assay later, we might see a much more striking effect. The problem that we haven't been able to figure out how to solve is that you have to use bortezomib to define this protein degradation as proteasomal, and this dose of bortezomib plus asparaginase is highly, highly toxic. There are basically no cells left by 18 hours. So we haven't figured out how to do this assay later, but it's at least consistent with the model that we're proposing here. So then we ask what regulates formation of these bodies. So you may know that the two best known effectors of amino acid starvation in mammalian cells are inhibition of mTOR and activation of GCN2. mTOR is inhibited in the absence of a few select amino acids, and GCN2 is thought to be activated by uncharged transfer RNAs, so transfer RNAs that are not conjugated to their cognate amino acid. And when you starve amino acids through these two factors, you induce this coordinated response through which consumption of amino acids is reduced by blocking protein synthesis, and you get an increased production of amino acids through lysosomal protein degradation and through an upregulation in biosynthesis and transport of amino acids. So we asked whether mTOR and or GCN2 would regulate these. And long story short, we found if you inhibit mTOR here with rapamycin, or if you inhibit GCN2 with an shRNA, in both cases, inhibiting these sensors is not sufficient to trigger formation of bodies, nor does it prevent the ability of these to be induced by asparaginase in both cases. So we think this response is actually independent of at least the best known effectors of amino acid starvation responses. So we then asked this question of heterogeneity, and this is a question a reviewer asked us, and it's a really interesting question. So, as I've showed you before, these bodies form, but they don't form in every single cell, and some cells form more than others. And this is quantified here. So we get, this sort of spectrum of some cells form a lot and some cells form a few. And so we tried to understand the biologic basis for this. So we generated a panel of single cell clones, populations derived from single cell clones. As you see here, we have some clones that form really no bodies at all in response to asparaginase, and others that form a ton. And I hope this projects reasonably well, where you could see that the clones that form bodies form really a lot, and it's more than in the overall population of the parental cell. And other clones have really none. This does not correlate with differences in protein expression of GSK3 alpha, so the levels seem to be the same at the level of a Western blot on the whole population. This does, however, correlate with sensitivity to GSK3 alpha inhibition with this drug, BRD-0705, plus asparaginase, with clones that form bodies being actually sensitive to the combination of GSK3 alpha inhibition. And interestingly, clones that don't form bodies are now totally resistant, suggesting that they've acquired, it's not that they're sensitive to asparaginase, it's that they've acquired an alternative mechanism of resistance. At least that's one possibility. But our efforts to study this have been stymied by the fact that resistance is not a stable phenotype. So we've applied genomics and basically several approaches that we could cook up, and we cannot ascribe resistance to something that we've been able to identify. And in fact, it's unstable over time, and suggesting it's actually not a non-genetic mechanism. We're very interested in understanding this, but this is still a work in progress. Okay, so now back to the role of GSK3 alpha body formation. So I showed you before that among non-essential amino acids, asparagine seems to be unique in its ability to stimulate formation of these bodies because it's unique in its ability to repress them when you add it back. But, proteasomal protein degradation, of course, is a catabolic source of all amino acids, at least all proteinogenic amino acids. And so it might be evolutionarily advantageous for this response to be triggered in response to starvation of essential amino acids that cannot be made by cells. And in fact, we found that if you culture leukemia cells in media lacking essential amino acids only, and now this media has asparagine and it has all of the other non-essential, but it's just lacking the essential amino acids, cells in fact form GSK3 alpha bodies. This is true not only in leukemias that presumably had a chance to evolve asparaginase resistance when they were in the patient, but also in normal cells. So if we take normal HSPCs and this is a cell line, so it's a bit of a stretch to call it normal, but it's derived from normal human intestinal epithelium, and we think it recapitulates at least some features of normal intestinal biology. And in both cases, we see that these cells in fact form GSK3 bodies when they're starved of essential amino acids. We then asked whether GSK3 alpha promotes survival during starvation using our favorite small molecule tool compound, BRD-0705, and this is a structurally related negative control. And as you see here, if we treat normal HSPCs or this cell line derived from normal intestinal epithelium, in both cases, inhibition of GSK3 alpha is toxic only when cells are starved of amino acids and not the negative control. So then we asked, well, is it every essential amino acid or are there just a subset of these? And so here the experiment is we're back in leukemia cells now, and we're assessing the ability of starvation of individual amino acids to stimulate this response, to stimulate GSK3 alpha body formation. And here are controls. Asparagine, of course, increases the number of bodies, and glutamine does not. We went through the whole panel of essential amino acids one by one, and we get this very striking result, at least I think it's very striking, that only two of these, leucine and valine, are able to stimulate GSK3 alpha body formation when they're absent, and none of the others. And I think the simplest explanation for this is that there are unique specific sensors for these amino acids, these three, asparagine, leucine, and valine, that signal deficiency and that sense deficiency and signal this to the GSK3 alpha and the ubiquitin-proteasome. And I'll get back to this in the next story. That we asked whether, sort of a clinical question, does formation of GSK3 alpha bodies predict asparaginase resistance in human leukemia? In fact, if you look at leukemia cell lines, GSK3 alpha bodies form in those that are resistant to asparaginase, much more than in those that are asparaginase sensitive. This is also the case in a panel of leukemia samples engrafted as patient-derived xenografts, which again, we get a very nice distinction between those that are resistant to asparaginase form GSK3 alpha bodies, and those that are sensitive do not. Moreover, if we compare PDX models collected at diagnosis from patients who went on to be cured versus those collected at relapse, obviously not cured after asparaginase-containing chemotherapy regimens, we see a much higher ability to form GSK3 bodies in response to asparaginase, arguing that they've evolved this as an adaptive response, or maybe it was pre-existing, but this adaptive response is responsible for treatment resistance, at least partly responsible for treatment resistance in these cases. Now, of course, it's nice to predict resistance, but it's not very satisfying if you can't do anything about it. So then we asked whether formation of GSK3 alpha bodies might predict sensitivity to asparaginase combined with a GSK3 alpha inhibitor. And in fact, this is the case. So GSK3 alpha body formation predicts sensitivity to BRD-0705, our favorite GSK3 alpha inhibitor plus asparaginase. This is true in cell lines and also in patient-derived xenograft models. And this, of course, has to be validated in an independent cohort, but you might imagine using this as a prognostic biomarker if we can ever cook up a clinical GSK3 alpha inhibitor in which to, with which to test this approach in patients. So our model at this point is that cells stimulate protein degradation through this pathway in response to asparaginase starvation by physically concentrating multiple distinct enzymatic factors that are all sequentially required for protein degradation through this pathway in GSK3 alpha bodies. This formation of these bodies requires HSP70 function, and we can maybe in the Q&A session, I'd be happy to get into some thoughts about why this is, although we have we haven't worked it out yet. Strikingly, formation of GSK3 alpha bodies is uniquely sensitive to loss of asparagine, leucine, or valine. And we hypothesize that there are one or more sensors for these. And again, I'll get back to this in the next part of the talk. So essentially, to conclude what I've just summarized in graphical form, I hope I've convinced you that GSK3 alpha mediates asparaginase resistance in a manner dependent on its N-terminal intrinsically disordered domain, that this domain mediates assembly of GSK3 alpha and the ubiquitin proteasome system, or at least multiple components of the UPS. The formation of GSK3 alpha bodies is specifically triggered by loss of only 3 amino acids, suggesting again that there are specific sensors for these. And we propose that formation of these bodies provides a mechanism to maximize the catalytic efficiency of proteasomal protein degradation in response to amino acid starvation. And I think one of the important conclusions from this is that this provides a potential explanation for evolutionary conservation of GSK3 alpha, because GSK3 beta, which, if GSK3 beta is really redundant, as is widely believed, and it's ubiquitously expressed, it's difficult to understand why there's been such strong evolutionary pressure on the maintenance of GSK3 alpha. So this provides at least one potential mechanism for that. Okay, so maybe I'll keep, well, I'm happy to take questions if you'd like, or I can keep going and we can save them all for the end. Maybe I'll just keep going. Okay, so the next question is how do cells, so, we're trying to basically find this sensor. Now, full disclosure, we're now getting to the part of the talk that is still work in progress, and we haven't completely worked out these mechanisms. I would very much appreciate thoughts and suggestions on this. So the question we're trying to get into here is how do cells actually sense asparagine starvation and signal this to GSK3 alpha and the ubiquitin proteasome system? Oops. And again, I'll remind you that this is really a strictly GSK3 alpha dependent effect that is not phenocopied by beta. So based on this, Laura had the idea that, maybe proteins that bind only GSK3 alpha and not beta, some of these proteins you would predict maybe are enriched in some of these sensors or effectors of this response. So Laura mined the database, the Bioplex database, generated by Steve Giggy and colleagues, where they've attempted to map the protein-protein interactome of every human protein, or at least every protein that they've tested, which is a lot of them. And if you do that and filter out proteins that are bound, so look for proteins that bind GSK3 alpha and exclude those that bind GSK3 beta, or that are known to participate in canonical Wnt signaling, which turns out to be a lot of these as well. And also another filter here, which is important, is by availability of antibodies, you end up with 4 candidates. Now, 2 of these actually are pretty juicy already, because this protein here is a ubiquitin binding protein that shuttles proteins for degradation through autophagy. And so at least that's what it's been proposed to do. So that obviously might have a role here. This other protein here is known to be a component of the Gator complex, which senses amino acid starvation in the mTOR pathway. And so while formation of GSK3 alpha bodies is distinct from mTOR, it's conceivable that there's some overlap in the sensors of the amino acids that are lost. And so these are interesting. But I'll draw your attention to PRN1, which there was no prior reason to believe this might have a role, but we focused on it because we got the cleanest result on pull downs, as I'll show you next here. So the first question Laura asked, do these proteins interact with GSK3 alpha? So the experiment is you take your leukemia cells and you do an IP for endogenous PRN with an antibody. And this is where the need for an antibody was an important component of our experimental approach. So at baseline, when cells are grown in complete media, there's very little interaction. So if we pull down PRN, we see very little GSK3 alpha bound. But if you starve the cells of asparagine before doing the experiment, now you get a very robust interaction. She then did the experiment the other way around, where now she's pulling down on endogenous GSK3 alpha and looking for binding of PRN. And again, we see little interaction at baseline, but a robust interaction when asparagine is removed from the media. This is true not only for asparagine, but also for the other 2 amino acids whose loss is sensed by our pathway. So both valine and maybe a little bit less, but also loss of leucine also stimulates interaction of GSK3 alpha and PRN. It turns out, not known to us when we initially started this, but there's some prior data suggesting that GSK3 beta can actually bind PRN. And Laura was able to validate the binding, as you see here, but this was asparaginase insensitive to asparaginase. So these bind whether or not you have asparagine. And so So this seems to be different than this role in canonical Wnt signaling. So what is prun? So this is a protein. The gene was first identified in a Drosophila mutant with prune-colored eyes, which explains its name. And initially it was proposed to be a GTPase activating protein and maybe to regulate RAS signaling. It has subsequently been shown to have several enzymatic activities. It's a phosphodiesterase that can inactivate the cyclic AMP and cyclic GMP second messengers. It's also been proposed to have, or shown to have, exopolyphosphatase activity. This, I knew nothing about this, but it turns out that bacteria, thermophilic bacteria, have polyphosphates, which are long polymers of phosphate. So sort of like ATP is a trimer of phosphate, but actually these are much longer. And they're thought, they're energy stores, much like ATP is, but they're also thought to directly regulate several enzymatic activities. And this has been described, well, really only in bacteria, and whether this is important in eukaryotic cells is really completely unknown. Prun also binds and interacts with NME1, which is a nucleotide kinase that basically forms GTP and other non-ATP nucleoside trisphosphate. So there's sort of a theme here emerging that this is some sort of a nucleotide signaling factor. There have been a number of studies that have implicated prun in really very different cellular phenotypes, but it's sort of a positive regulator of many oncogenic-like phenotypes. So signaling, proliferation, migration, differentiation, metastasis. So we asked, does prun1 interact with amino acids? So we did a pull down of prun1, endogenous prun1 from human cells, and that's an important point to note here. And now we're going to take this purified protein and incubate it with fluorescently labeled amino acids. And we have asparagine, which we predict should bind, at least we know that this regulates our pathway, and glutamine, which I'll highlight is very closely related but does not. We find that prun1, again, when purified from human cells, binds asparagine quite well and not glutamine. So that is consistent with our role in our pathway. We did the same experiment with valine, leucine, and isoleucine. And I'll draw your attention to the fact that these are closely related to each other. These two stimulate GSK3 alpha body formation when they're absent, and this one does not. And so again, we see binding to valine, binding to leucine, and not to isoleucine, which all fits. Now, the thing that's difficult for me to understand, how a single protein, I mean, if this is one domain, one binding site, how can you distinguish asparagine from glutamine and valine from isoleucine, but bind both valine and asparagine? If that makes sense, it's a little bit difficult to believe. And the caveat here is we're pulling down the protein from human cells. So I wonder whether we're pulling down cofactors that actually are the direct amino acid binders. We're in the process of making a recombinant protein now to test that prediction, whether that's the case or not. And so this is all, as I mentioned before, still a work in progress. We then asked whether prun is required for formation of GSK3 alpha bodies in response to amino acid starvation. And here are our control cells. As I showed you before, when we deplete either asparagine, leucine, or valine, we stimulate formation of GSK3 alpha bodies. And if we knock down prun, we basically completely block that, or almost completely block that. And we then asked whether prun actually moves into GSK3 alpha bodies in response to starvation. And in fact, when we starve cells of asparagine in this experiment, treating with asparaginase, we see that prun1, in fact, co-localizes with GSK3 alpha in what we believe are the same GSK3 alpha bodies. This is quantified here. And then we asked whether prun1 is functionally required in order to tolerate amino acid starvation. So here we're doing a genetic knockdown with shRNAs. And in fact, if we treat cells with asparaginase, again, these are pretty resistant at baseline. We knock down prun, and this basically phenocopies loss of GSK3 alpha, consistent with the model that this is working in the same pathway. This response is unique to asparaginase. It is not triggered by any, in other words, loss of prun does not change sensitivity to any other chemotherapeutics that we've tested. And we can rescue this phenotype. So of course, an important question whenever you do an experiment and death is the readout, you have to make sure it's an on-target effect. And so here the experiment is we're knocking down prune. You can see that we induce toxicity only in the presence of asparaginase. And this is completely rescued when we restore wild type prune. I told you before this has enzymatic activity, and in fact, there are both drugs and point mutants described that disrupt its enzymatic activity. I'm not showing you any of that data though, because we're getting discordant results. And so the small molecule inhibitor and the mutants that are supposed to be loss of function don't phenocopy each other. And so either the drug is killing cells through non-specific toxicity, or the point mutants are actually not loss of function, and we're still trying to work out whether or not this is an enzymatic dependent effect. We then asked, the most conclusive experiment, in my opinion, that would demonstrate a role for prune-1 in this pathway is if we can rescue loss of prune-1 by reactivation of a downstream factor in the same pathway. And here we took advantage of the fact that actually there are open gate mutants of a proteasomal subunit, PSMA4, that we know are in GSK3 alpha bodies. And when you express these in cells, this is basically a deletion of the N-terminus that results in the formation of proteasomes that are hyperactive for degradation of multiple substrates, maybe all their substrates. So the idea now is we're going to test whether loss of prune-1, this toxicity can be rescued by directly upregulating proteasomal function. And in fact, we found this is the case. In our leukemia cells, as I showed you before, when we knock down prune, they become sensitive to asparaginase. And this toxicity is completely rescued when we put in this hyperactive proteasomal mutant. So I think this is the strongest data that this is actually functioning in the same pathway. So in conclusion for this part of the talk, I hope I've convinced you that prune-1 binds to GSK3 alpha in a manner dependent on loss of select amino acids, which are the same ones whose loss stimulates GSK3 alpha body formation. Prune not only both regulates this pathway and it's required for the formation of GSK3 alpha bodies. The toxicity of prune can be rescued by directly stimulating proteasomal function. And so we propose that prune-1 functions in a pathway that senses amino acid starvation and signals this to upregulate protein degradation. We're still trying to sort out whether this is actually the sensor for one or more of these amino acids, or is it a signal transduction molecule downstream of the sensors? And so there's a lot of work to be done here still. So this is the end of that part. And then in the last few minutes, I just want to give you a flavor for the latest, which is work of Yunchil Che in the lab, who basically was interested in understanding or in developing new approaches to trigger asparaginase sensitivity. So he came to the lab interested in Laura's paper in colorectal cancer. His background is in colon cancer. And basically trying to find other ways in which we might be able to sensitize APC mutant tumors to asparaginase. And maybe I'll just skip ahead. I'm just cognizant of the time. I want to leave plenty of time for questions. I'll just skip ahead a little bit. So the screen that Yunchil did was in an APC mutant colon cancer cell line. Again, it's a loss of function genetic screen. And the comparison is treating cells with either vehicle or asparaginase. And he's looking for CRISPRs, or genes whose loss is not toxic on its own, but it's profoundly toxic when you lose asparagine or when you treat cells with asparaginase, if that makes sense. So here are the results of his screen. And I always like to make sure to convince myself the screen worked by finding internal positive controls. And we have a whole series of them here. We have asparagine synthetase, as well as multiple proteins that are known to function in the amino acid starvation response in response to this factor GCN2 that I mentioned before. So GCN2 is here. GCN1 is a cofactor required for GCN2 activity. And it works by upregulating ATF4, which is also here. And one of the important effects of this is an upregulation of ASNS. And so this is, I think, in my mind, very convincing that the screen worked quite well. There was one gene among the list of hits that we had it was very difficult to explain its presence here, which is G6PD. And of course, this got our attention. It turns out, so before I get into the G6PD, and again, this is all work in progress, and I would love to get into this perhaps in the question and answer session, but The first thing Childr did is validate that knockdown of G6PD in fact induces asparaginase sensitivity. And you can see here the genetic knockdown, which you get actually very remarkably good knockdown, it induces profound sensitization. Oh, I'm sorry. This is the activity. This is validation of shRNA knocks down the RNA level, also knocks down activity of G6PD. And we get significant sensitization to asparaginase in colorectal cancer cell lines. The effect size here is very similar to what we saw with GSK3 alpha inhibition. Importantly, this induces asparaginase sensitivity in colorectal cancer cells, but not in this cell line derived from normal human intestinal epithelium, which of course is an important consideration when you're trying to kill cancer cells. It's easy to do if you don't worry about not killing the patient. So this gives us some confidence that there's going to be a meaningful therapeutic index here. There are, there are G6PD inhibitors that have been used for many years in literature, and actually this beautiful paper of Josh Rabinowitz and colleagues showed that actually those inhibitors are not inhibitors of G6PD in cells, but they do describe actually a new inhibitor here, which is called G6PD-I1, shown here. This is really a tool compound. It does not have favorable features for in vivo therapeutics, but you can use it in cell lines. And we see that the small molecule inhibitor phenocopies the genetics. So we get sensitization to asparaginase in colorectal cancer. And finally, Childr has moved into organoid models now. So we have this organoid model of APC mutant colorectal cancer I showed before. These are mouse intestinal organoids engineered to sort of precisely phenocopy the mutations of human colon cancer. These organoids are, if you treat them with a G6PD inhibitor, basically nothing happens. They're pretty resistant to asparaginase, but if you put both in combination, you get this really profound toxicity. And this is in vitro, of course. We're working to bring this in vivo as we speak. And the question in our mind, and this is really an open question still, we haven't figured this out, how does G6PD loss induce asparaginase sensitivity? So you may know that G6PD catalyzes the rate-limiting step of the pentose phosphate pathway. This pathway is important for many things, and you might predict that many of them conceivably could induce asparaginase resistance. So it's a major pathway through which cells regenerate NADPH. And NADPH powers a number of enzymatic reactions. So it's a redox factor to handle reactive oxygen species. It's involved in the biosynthesis of multiple metabolites, especially fatty acids and sterols. Of course, ROS or either of these, one or both of these could be involved. And there's also the non-oxidative pentophosphate pathway, which is required for the production of nucleotides, especially RNA metabolites. And so we're working through this. At the moment, we don't think this is, we think this is probably an NADPH dependent effect, but actually it's quite difficult to rationally find an enzymatic reaction powered by NADPH that you would predict is involved in asparaginase biosynthesis or in proteasomal degradation. So we're actively working on this, and I hope to have an interesting answer in the near future. So I think I'll stop there and make sure to leave plenty of time for questions. So for this last part, we've shown that a genetic screen revealed that loss of G6PD induces asparaginase sensitivity in APC mutant colon cancer. I didn't show the data, but actually this is also effective in leukemia. Genetic or pharmacologic inhibition of G6PD induces asparaginase sensitivity in distinct models of colon cancer. We believe the effect of G6PD loss is mediated by depletion of NADPH, and we're quite interested in defining what is the downstream mechanism. So I'll stop there. Lots of people to acknowledge. Really, Laura and Childr have driven most of the work you see here. We've gotten some help from many collaborators. And of course, thank you to the funding agencies, and I'm happy to take any questions.
Alejandro, just fascinating work, and I think a beautiful mechanistic dissection of the things you're interested in. Really well done. Let me start with questions that are in the chat box. Jun Yang says, great data. And two questions. First, what do you think is the functional consequence of the GSK alpha body formation? Does it increase GSK alpha activity, stability, or something else? And two, there's a wide variation in asparaginase sensitivity in ALL patients by subtype. And do you think that common genomic lesions seen in ALL might directly influence GSK alpha function, such as ETB6-RUNX1?
Right. So for the first part of the question, does GSK3 alpha functionally change when it moves to bodies? And the short answer is we don't know. The model of phase separation or biomolecular condensate formation is that you can stimulate the overall rate of a multi-step biochemical reaction by putting all the enzymes together so that the products immediately feed from one enzyme to the other. And that's what I've been assuming happens. But of course, it doesn't. This is not mutually exclusive with the idea that something happens in those bodies to change GSK3 alpha activity. We've done sort of a rudimentary look. There's two known phosphorylations on GSK3 alpha that are known to regulate its activity, and they don't change. But of course, that's only two out of probably an infinite number of things that could change. So interesting idea. We don't know. And for the second part, could oncogenic mutations change response to this pathway or change dependence on this or alternative sources? And in fact, I think actually that's the case. In the context of colon cancer, we found that the reason colon cancer tumor cells are sensitive to asparaginase when you activate this Wnt branch, and normal cells are not, or normal cells, again, this is the cell line that we like to use, is because normal cells can rely on autophagy as an alternative catabolic source of amino acids. And this response is actually blocked, at least in colon cancer, by KRAS and p53 mutations. This has been previously described by others. And I think it's not a coincidence that actually the leukemia cell lines that we've been working with, by coincidence, quote unquote, are all KRAS and p53 mutant. So there may be something very special about this genotype, which is of course a common genotype in human cancer, but as you know, there are many leukemia subtypes that don't have these mutations. And so whether that'll pan out as an important determinant, we'll have to see. I would suspect it might. And I'm sure it's also not the only, they're not the only oncogenic factors that will regulate response to this pathway. So lots of work to do there. Very interesting.
Marius Kelp says, really impressive data, exclamation mark. I might misunderstand, but why is a selective GSK3 alpha inhibitor needed to overcome asparaginase resistance instead of available proteasome inhibitors such as bortezomib, carfilzomib, or others? Yes,
great question. And you may know actually Dave Tichy and COG have run a trial of bortezomib in TLL with 4-drug induction. The trial, I actually don't know all the details, but I think it is probably underpowered to answer the question of whether this improved outcomes. And I don't know why I think it was stopped early, and I'll have to ask Dave Tichy at some point. But in fact, if you treat cells with proteasome inhibitors, you profoundly sensitize to asparaginase. But actually the difference is that's also quite a bit more toxic to normal cells. If you inhibit GSK3 alpha plus asparaginase, actually we see almost no sensitization of normal cells to asparaginase. But the proteasome inhibitor actually does increase the toxicity. And so, excuse me, the sort of clinical reason I would personally not dive to proteasome inhibitors. I think the therapeutic index is likely to be quite a bit worse. And the biologic implication of that is, cells may rely on alternative routes of proteasomal protein degradation to tolerate this that may also be impaired in the tumor cells. And so, what we're interested in that, of course, but we have no particular insights on that at the moment.
Great. Yuji asks, very nice talk. Asparagine and glutamine structures are quite similar. Do you know why only asparagine has an effect?
Yes, excellent question. Presumably, there's a sensor that has evolved to be highly sensitive to asparagine and not glutamine. And why that would be, I don't know. I mean, I could speculate for a long time probably, but we just don't know. I think the simple explanation is that there must be a sensor that distinguishes between these two. But why that would have evolved, we're interested in that. I just don't know. Great.
So let's see. Question. Michael Schwab says, is BRG0705 selective for GSK alpha over GSK beta, and is the binding site known?
Yes. So GSK3 alpha, I'm sorry, BRD0705 is sevenfold selective for alpha over beta, which is, on the one hand, you would say not super impressive, but actually the ATP, these are both, or most, maybe all GSK3 alpha inhibitors are ATP competitive. And the ATP binding site of alpha versus beta differ by a single amino acid. So actually, I think it's actually relatively impressive. Um, the specificity of alpha, I think, is important because if you inhibit both GSK3 alpha and beta, that leads to beta-catenin activation. And there's evidence from other inhibitors that if you do that in model systems long enough, you end up getting intestinal adenomas. So benign tumors of intestine and also of the bile duct system. And so there's a concern that if you do that long enough in patients, you will eventually stimulate colon cancer. So I think increased specificity would certainly be beneficial, but if you had refractory leukemia and you were talking about doing this for 3 months, I would suggest that maybe the therapeutic is just fine for a pan GSK3 inhibitor.
Great. We're getting close to the top of the hour. So I do want to thank Dr. Gutierrez for an excellent talk, but also note that he has agreed to stay on past the top of the hour for extended questions and answers. And so just before we close the formal part of the event, but continue on, I encourage people to stay on for the questions. I want to thank him for speaking and note that next week, April 14th, we have Dr. Yataka Yasui of St. Jude presenting on the pathobiology of chemotherapy resistance in T-cell acute lymphoblastic leukemia. The typo there on my part. What can we learn from GWAS data? Genetic heterogeneity and rare variant hypothesis of cancer risk. The week after that, Dr. Susie Baker will be presenting on her glioma work. Okay, so returning to questions. Have you done mass spec analysis for GSK3 alpha under asparaginase depletion conditions to see what's happening and who its friends are?
Yes, in fact, a reviewer asked us that question, and we did that. If you did pull down, so we did two things. One is we pulled down endogenous GSK3 alpha with and without asparaginase. And basically, we see no differential binding partners. And in fact, we find no members of the proteasome that we know co-assemble. That's consistent with predictions. This whole idea of phase separation is that it's mediated by low affinity interactions that are not stable to IP. And I think that's at least consistent with that model. Now, of course, you can never be sure that you didn't just, there's some technical failure there. But we find GSK3 alpha binders, but none of them are obviously involved in our pathway. We also did try proximity proteomics with BioID, and actually, when you fuse a biotin ligase, at least the way we've done it, to GSK3 alpha, it no longer localizes properly. So that will need more work. We haven't quite figured out how to do that.
Excellent. People, you can continue to type in questions in the chat box. You raised during the talk about why would mammals care about asparagine in a way that other vertebrates wouldn't? So let's return to that. What do you think is going on there in the fascinating mechanism that you've begun to propose?
Yeah, so that's a really interesting question. I cannot think of another cellular response to starvation that's not conserved back to yeast. mTOR, GCN2, all these things are really evolutionarily ancient. And while it's possible that other organisms, other vertebrates, can stimulate the same response using a different domain of GSK3 alpha, for example, I think it raises the very kind of provocative idea that actually mammals have evolved a new starvation response. And so why would that be? So we're making knockout mice right now where we basically replace the key domain of GSK3 alpha with that of beta, that I hope will give some insights into this process. But this is all pure speculation. But one idea that comes to mind: the asparaginase we use clinically is from E. coli, and E. coli, as far as I know, is a mammalian-specific intestinal bacteria. And could it be that we've evolved this whole pathway so that the intestine can tolerate E. coli that are secreting asparaginase, And you might ask, well, why would that be beneficial? Well, maybe that's one of the ways that E. coli can clear out all the pathogens from your intestinal tract, C. diff and whatever else that you get. So that's one possibility. Another possibility comes actually from the work of Chris Walsh and colleagues, where they are sequencing, they're children with focal epilepsies that are due to cortical malformations. And if you sequence those patients have somatic mutations of some amino acid sensors in the mTOR pathway. Could it be that actually as the mammalian brain grew, you had to evolve additional mechanisms to tolerate asparagine starvation? Maybe it's particularly rate limiting. Again, pure speculation, but maybe these mice will get seizures, or who knows. And a third possibility is that another difference between mammals and most other vertebrates is most vertebrates, their early embryogenesis is in an egg with a giant nutrient supply. And could it be that actually in mammals, where the yolk is actually quite small, we've evolved this whole strategy to survive maternal starvation, maybe in the pre-implantation period? So those are all questions we're hoping to ask with these mice that'll be coming at some point.
Great. Blanca Dietz asks, do you have any idea if the mechanism could be the same in Wnt mutant medulloblastomas?
Yeah, so as far as I know, I'm no expert on medulloblastoma, but I think the mechanism of Wnt activation in the Wnt positive medulloblastoma, I think it's not known, and it's often not mutational. Now, maybe you'll tell me that's completely wrong, and I have to admit, I haven't read this literature in a few years now. But it certainly could be the case. Now, there's a bit of a problem that asparaginase doesn't cross the blood-brain barrier so well. But actually, people have tried intrathecal asparaginase for CNS leukemia. So if the mechanism is overlapping, there would definitely be a route to testing this approach. And it may actually be worth testing. I don't know if there are cell line or organ models of Wnt versus other medulloblastoma subtypes. That may be an interesting avenue of investigation there.
Yeah, it could be. It's interesting to think about. There are people, both St. Jude and Farber, who are interested in the medulloblastoma groups. And you're right. It's not that she didn't write mutant. It's the Wnt subgroup that could be looked at. Xuan Yu asks, I think does the GSK3 alpha inhibitor, is it used in the clinic? I think you said not.
It's not. The drug we use is a tool compound, not. We're trying to get a better drug to the clinic. It's been sort of slow going, but we're still pushing on that. And I hope we'll have something before I'm ready to retire, but it's not happening right now. I mean, we're trying.
Great. George Chen asked a question early on. You addressed this partly, but is the drug similar to proteasome inhibitors such as Velcade for killing multiple myeloma cells? What's the similarities and differences in terms of the drugs and mechanisms?
Yeah, good question. We haven't looked at that. I had never thought of that before. I mean, it might be. I think my maybe naive idea is that in myeloma, you just are making so much immunoglobulin that you need kind of basal protein degradation to prevent all these aggregates. Now, as I say that, actually, part of the factors we found here are like HSP70. We found that because it's involved in degradation of aggregates, so there could be some overlap. It's actually, yeah, it's worth testing for sure. Excellent.
So I want to ask a question. In terms of the targets of protein degradation, do you have any? Is this random proteins that are just grabbed, or might there be stores of proteins that are highly enriched for essential amino acids that actually, in a way that fat acts as a store of lipid, these things could act as stores of the essential amino acids? Any data on what the targets are?
Yeah, so we've tried that with, not in a very sophisticated way, but we just did global proteomics on cells. And we don't find any individual proteins that are differentially degraded, but there's a big caveat to that, which is that our approach is just not that sensitive. And so you're basically measuring abundant proteins. I mean, I think it's always a problem with proteomics that you're always biased towards abundant protein. It's very difficult to rule out a less abundant substrate. So I don't know. I favor, I guess the way we've been proposing that this happens is that this is somehow global protein degradation. One hypothesis that actually links back to HSP70 HSP70 is best known as a protein folding factor. It's also been described to unfold damage to proteins for degradation in this pathway I mentioned. And so could it be that one way that this response is regulated is by activating the unfolding activity of GSK3 alpha? And in so doing, you're now revealing GSK3 alpha consensus phosphorylation sites that are normally buried in the center of a protein. And that's one idea. There's only a single experiment I could think of to test that. And so I have been a little hesitant to try to convince a student or somebody to dive into that because I'd like to make sure we can get a whole paper out of it as opposed to one experiment. But it can be done. We just need to think a little harder and maybe talk to people who know more.
Great. We have 2 more questions. People can feel free to add additional questions into the text box. You talked a bit about normal cells, so it's interesting to think about therapeutic index here of a model of depleting an essential amino acid, stimulating then protein degradation, and why that is not toxic in normal cells. You talked about autophagy. Have you actually tested that in any way to find out how the normal cells are escaping this?
Yeah, so this is actually, we've done that both in this cell line that I mentioned, derived from normal intestine, and also in mouse intestinal organoids. And in both cases, if you treat them with asparaginase plus autophagy inhibitor, actually that's toxic. It's quite a bit more toxic than GSK3 alpha inhibitor and asparaginase. So at least in those 2 models, I think our data is pretty strong that autophagy is important.
Great. Can the N-terminal domain of GSK3 alpha be used to pull other proteins into this body? So is it, can you use it as a targeting domain?
Yeah, good question. We haven't tried that. That's an interesting question. We have done one experiment, which is we fused the N-terminus to halotag, which is this domain that you can actually image in live cells. And actually, it does work. You can pull in halotag into those bodies. So yeah, you could think of pulling other things and then trying to manipulate this. That's an interesting idea.
Yeah, it's interesting to think about the fundamental mechanisms that are underlying the association of these proteins together. That's great. I want to thank you, Alejandro. Really, I can't say enough. Fascinating mechanistic work and just a beautiful dissection at a level that I think is really not commonly seen. So thanks. One more just popped in. So the question is, can you detect GSK alpha by using mass spectrometry of blood?
Yeah, you can. There's a few different ways. There are paralog selective antibodies. So the more commonly used antibodies detect both. And so you may notice from papers, or if anybody is on Westerns, you'll often see the 2 bands separately. So you can detect them by molecular weight on a Western. There are paralog selective antibodies that you can use. And I'm sure mass spec would work too. We haven't, well, no, that's not true. We did find GSK3 alpha and beta in our mass spec experiments. So I'm sure that would work. And thanks, Charlie. This is really, as I said, this is really one of my favorite seminar series. And on the one hand, I'm eager to go back to in-person seminars. On the other hand, it's so great to be able to just keep up with everybody's exciting work, without having to necessarily travel. So it's really, really an honor.
Fantastic. Thanks again, Alejandro. And for everybody else, we'll see you next week. Take care.